So here we're going to look at chapter 6, cellular respiration. We're going to go through the whole thing, kind of start to finish, to kind of wrap up everything and give you a general overview and then the details that you need to know to work through class this coming week. So let's go ahead and get started. So we want to talk about how we are looking at aerobic respiration. We're going to look at the stages, and then the other two we will actually cover later and not in this video. So cellular respiration is an aerobic harvesting of energy. So aerobic requires oxygen, and that's what we want to look at in terms of what happens when you have oxygen and you are in good standing. So we know that life requires energy. In almost all ecosystems, energy comes from the sun. We've talked about photosynthesis and how that already works. So really what we're going to look at here is how to undo what photosynthesis has done. So we want to see how that process takes place. So in cellular respiration, the big thing to remember is that sugar is broken down into carbon dioxide and water. So we know that equation, and the cell captures some of that released energy to make ATP. Is what we want to look at. So thinking about our equation, so we're going to take C6H12O6 plus 6O2, and that's going to yield 6CO2 plus 6H2O. So that's what's going to happen. Energy and electron buses and all that stuff are in there, but that's what has to come in and that's what has to go out. So as we put cellular respiration on the plate, we can't really forget about photosynthesis. These are a cycle that basically work through matter and energy cannot be created or destroyed. So the matter is just cycled through these two processes, and then obviously we have an input of energy and an output of energy as well. And that's what we're going to kind of look at. And what we're going to do is cellular work. So let's just talk a little bit about cellular work real quick. So there's three main ways of cellular work. We're going to have mechanical. So that's what we use to move things, our body, our heartbeats, things like that. We're going to have chemical. Okay. Because chemical processes do require some input of energy. We've seen that with our enzymes. Enzymes lower the activation energy, but you still have to put in some energy to get energy out. Enzymes just allow a little less. And then the last one is transport. So we've seen that with like active transport is the primary example there. So those are the three types of cellular work that cells are going to do to make things happen. So that's what we need energy for, is to do those three types. So respiration, we have to keep in mind that there are two sides to respiration. So there is the breathing and then the cellular respiration. So breathing and cellular respiration are not the same thing. They go hand in hand and they work together, but they're not the same thing. Respiration in the breathing sense refers to gas exchange. So you inhale and you exhale. And your lungs bring in oxygen and send out CO2, and that gets changed within the bloodstream, and that's what breathing is. The actual cellular respiration is an aerobic harvesting of energy. So that's the chemical processes that we looked at. We've talked about glycolysis, the Krebs cycle, and electron transport chain and oxidative phosphorylation. So that's cellular respiration. You need one to have the other. And so it's important to understand that relationship that they build. But they are not the same thing. So breathing and cellular respiration are not the same thing. So nice little diagram that kind of talks about that. So again, breathing up here, O2 comes in, CO2 comes out. So this right here is just the breathing part. It's transported in your through your lungs into the bloodstream, and then you actually have cellular respiration that's occurring in the mitochondria or in the cell. In the case of glycolysis, that creates that and makes that happen. So big things of cellular respiration, things to think about. So it's an exergonic reaction. It's going to release energy from that glucose in the form of ATP. That's what we want to finally get to. We're going to produce, here it says 32. Again, I said anywhere, I said in class 34 to 38. Now you might actually see 32. Again, it's all about efficiency. As long as you're in the 30 mark, you're okay. It just really depends. And it uses about 34% of the energy originally stored in glucose. The rest is lost as heat. So really, this number is all about efficiency and how efficient capturing that energy is and converting that energy. And so you get all kinds of numbers. Anyway, like I said, now it's 32 to 38 in that regard. So you've seen this equation before. It's just the reverse of photosynthesis. So sugar, oxygen gives us carbon dioxide and water, and then we do generate some ATP and some heat energy as well. So your body requires a continuous supply of energy. There's always work going on in your body. Your heart is always beating. Your blood is always moving. Your brain is always functioning, diaphragm, all that sort of stuff. So cellular energy work is always going on. And so we have to balance that work with some things to think about. And so this is just kind of like that application base, like why should I care about cellular respiration? So a little bit about, ins and outs. Normally I hop on a soapbox about diets at this point. They're not really effective in the long run. It's more about your eating habits and in and out. So counting calories and looking at that process, matching your ins and outs. But just to give you kind of perspective of how much calories you can burn doing various things. So just by taking notes, you're burning 28 calories an hour. If you're running an 8 to 9 minute mile, you're burning almost 1,000 calories in an hour. So more exercise, more energy you burn, more you got to consume. So just as a quick reminder, we're kind of working backwards. So remember, Leo says GER. So this is kind of backwards from what photosynthesis did. So glucose is going to become oxidized. Okay. So we're going to lose electrons. As I mentioned in class, we like to now talk about hydrogen atoms a little more. I don't really jump on that bandwagon. So we're going to lose electrons, and there in turn gives us CO2. Our oxygen is going to gain electrons. Remember, it's our final electron acceptor. So it is being reduced, and that becomes water because it has those negatives and it wants positives, so it finds hydrogen. So again, oxidation reduction reaction, Leo says GER. You can't have one without the other. Okay. So this, not super important to know, but just kind of remember the idea that oxidation reduction happens. So our buses are gaining electrons, so it's being reduced, and it does that. I just kind of want to point out that we do have an enzyme that does that, because remember, nothing happens in your body without an enzyme, a protein especially, but enzymes, all chemical reaction. So adding those electrons on, things like that happen. So this is kind of just a little bit behind the scenes of what's happening to how we load a bus. We've kind of talked about these buses as we go through. So stages of respiration. And I know I'm going kind of quick. You can always pause and rewatch and things like that. I'm just trying to hit the highlights and making sure we're understanding the basics of our notes. So we're going to break this down. Remember, there's the 3 stages that we looked at in the Poggle. This is an amazing picture. I would highly recommend, just like we had that chloroplast picture, this is the mitochondrion picture. So I highly recommend starting with this picture in the center of your paper and drawing out from it. So we've got our mitochondria here. And so here's our cytosol, or cytoplasm, that's outside the mitochondria, so inside the cell. So glycolysis happens here. Glucose is converted into pyruvate. That's going to generate a few ATP, not a lot. Okay, we're going to fill in numbers later. It's also going to give us some buses. So there's a bus that goes this way. Honk, honk. Okay. There is a stage that we didn't really talk about in the Poggle, but that's the pyruvate oxidation stage. Based on that name, you could think what that means. It's going to give some more electrons. The citric acid cycle. So congratulations. It's got two names. So the citric acid cycle is also called the Krebs cycle because Dr. Krebs discovered it, but it's called the citric acid cycle because it has citric acid in it. Okay, so tomato, tomato, potato, potato. Now, if you notice, here's where we lose our CO2s. This oxidative of pyruvate gives us some CO2. We get some ATP and some more electrons. And then oxidative phosphorylation kind of comes into play with the electron transport chain, and we have oxygen that picks up the electrons, makes some water, and generate a whole bunch of ATP. So that's what we want to focus on. So that's the overview. Okay, let's dive into a little bit of the details real quick as we go through. So I'm going to kind of these couple slides are just kind of the overview. If you know these, take that picture and put these together. And that's basically what I'm going to do in the next several slides as we go through. So stage 1 is glycolysis. So let's have our checklist here. So first thing to know is it occurs in the cytosol. So that is the location. This is the start of cellular respiration, so stage 1. And really the idea is we're going to break down glucose and make a 3-carbon compound called pyruvate or pyruvic acid. So it just depends on where we know it at that point. Okay. Remember, we are going to make a few NADHs and some ATP as well. So we'll talk about those a little more in detail as we get there. Stage 2 is the pyruvate oxidation and the citric acid cycle. We generally lump those together, and so the Poggle kind of did that as well. We just didn't really talk about that. It does now take place in the mitochondria. So there's your location. Okay. And what we're going to do is we're going to oxidize pyruvate into a 2-carbon compound. Okay, so that's the part we didn't work through. And then what that's really going to do is supply the 3rd stage with electrons. So we're going to load up our bus as we go through with that. And then eventually we're going to break that 2-carbon compound into a single carbon, and that's where we're going to get our CO2 out. That's going to come out of all that. So that's kind of the idea there. And then finally, stage 3. Stage 3 is completely nothing new to you. So this basically is just like photosynthesis. So it takes the same playbook out of there. Oxidative phosphorylation, chemiosmosis, all that is oxidative, is in the electron transport chain. It all kind of happens. So NADH is a bus, FADH2 is a bus. They bring electrons in. That is going to move that hydrogen ion against its concentration gradient. Most of the ATP is created here due to ATP synthase. And then our oxygens are finally an electron acceptor that picks up the water, picks up the electrons, and then turns into water. So that's what we've got there. So real kind of take that picture, put all that together, look through that. I'm going to break it down a little bit more into each step. So again, here's that wonderful picture again. I highly recommend focusing on this, expanding out almost like a concept map and thinking about these things that are going on in the different regions. So let's hit glycolysis real quick. I want to break these down. So ATP is used to prime glucose. Again, as I mentioned, we talked about enzymes. In order to make money, you got to spend money. So the first thing that we have to do is we have to add some ATP. Okay. So you got to put some energy in to get that molecule started. So just like you want an explosion to happen, you want a firecracker to go off, fireworks, you've got to light it first. Okay, and so that's going to create these 3-carbon intermediates that are oxidized, and we get our 2 molecules of pyruvate. Okay. We're going to net out 2 ATP, which means we actually make 4 ATP. and 2 NADHs. Okay. Now, just so you hear these words, ATP is formed by what's called substrate level phosphorylation, which is basically a fancy way for saying that there is no ATP synthase. So it's kind of happening at the substrate level, so it's kind of happening, it's kind of like a backdoor. It's kind of how you kind of can make some ATP without having the actual maker itself. Kind of a good analogy of this would be like the way you might have made ice cream in chemistry using like the rock salt and the coffee can, if you were here for that, versus actually having an ice cream maker. So, you know, you can kind of backdoor some ice cream very simply, but it, you know, you may not get as much ice cream or as good ice cream without using the ice cream maker. So this is what you need to know. This is what I would write down. This is a great visual for those visual learners. I've got my glucose, so 1, 2, 3, 4, 5, 6 molecules of molecules of carbon, atoms of carbon. I'm going to get out 2 ATP. I'm going to get out 2 NADPH, and then I have 1, 2, 3, 4, 5, 6, my 2 pyruvate. The only thing to add to this, to make a note, is, you know, you may see some questions on there. There is an investment of 2 ATP, okay, and we actually gross 4 ATP. So we're actually going to get out 4 ATP, but because you put in 2, you get 4 out. You always want more return on your investment, so you end up with only 2 ATP at that point. So these slides are going to get kind of muddled, so I don't want you to really worry about these. I'm just going to kind of hit through the highlights. You can kind of just follow along, but I just want you to know where we're kind of at. Always, this is the picture. This is what I want us to focus on. These are pictures in your book. They're nice because they kind of follow the steps. So here's our investment phase. So we're adding that ATP in, doing some weird things, adding some phosphorus to the glucose molecule, right? So glucose goes through and we make those steps, and then we eventually break it into voila. And the reason I want to talk about this is we make G3P. Remember, we talked about in photosynthesis that photosynthesis actually made G3P out of the Calvin cycle, and G3P went on to make sugar. Well, this transition from glucose to G3P, that can happen with any sugar at this point. So if you had fructose or galactose or sucrose or maltose or all the oses, this process has to happen first to get it to G3P. G3P is that molecule, that monomer that all sugars are based out of. So what we have to do in the beginning is get it back to G3P, and then we go from there. So that's kind of that idea. So what we're going to do is after we've done that, we've got it back to after we got it back to the G3P, we're going to rip off some electrons. And anytime you break that bond, you rip off those electrons, we're going to get some energy back out as well. So that's going to happen. And then we do get a little water out here. You're going to see a little water coming out in quite a few different steps as we go through. I'm not really concerned about where the water individually is coming from. There's a lot of water. We're made up of water. And if you notice, we get another set of ATP. So again, we put in the 2, we get out the 2, and voila. Okay. So, but I just want you to go back to this. So I know I said a lot there. Those are just some very details. There might be some things in there that would push you from a B to an A if I remembered that, you know, we went to converted to G3P, or we had that, you know, we actually made 4 ATP, but we only netted 2. But if you know this right here, then I would say you're pretty solid. I wouldn't stress over the other things. There's more important things to kind of know. So now what about this weird oxidate? So after I make my pyruvates, I've got to do something else now. And so the oxidation of pyruvate yields acetyl-CoA. Okay. A CO2 and an NADH. So we like to call this the kind of like a prep stage. The G3P, when it gets converted into pyruvate, the pyruvate is not quite ready to get into the citric acid cycle or the Krebs cycle. So there has to be some work done before it gets into the cycle. And this is what happens. So there's that stage there. We'll talk a little more about that. And then as the citric acid cycle turns, and remember, it says each turn, because how many pyruvate do we have, or pyruvic acid? 2. So this is 2 times, right? So 2 carbons from acetyl-CoA are added, 2 CO2 are released, we get 3 NADH and 1 FADH2. So if I multiply that by 2, I'll get up here, so just to kind of keep our numbers, 2 CO2, 2 times 2 is 4, 4 CO2, 4 and 2 is 6 CO2. Check that our 6 carbon. I'll get 6 NADH there and 2 FADH2 there. But I've also got, because you're like, oh, that's not what the Poggle said, huh? Okay, there's 2 NADH there. So 6, 7, 8 NADH there. Okay. So that takes care of all our numbers from the Poggle. If you go back and look at your Poggle sheet and look at our sum of all our ATPs and NADHs and FADH2, all that should make sense at that point, and where you're going to be. So this is what it looks like. So this is the citric acid cycle. This first part is the prep cycle. So we take pyruvate here. We do some chemistry to it. We'll get out a CO2, but remember this all is happening twice. So we'll get CO2 out of that. We'll get a bus out of that. And then what we actually get is what's called acetyl-CoA. So we've got 1 carbon, 2 carbon, and this CoA. So let me talk about this CoA guy for a second. So I like to think about the CoA guy as a name badge, a security tag. So a security badge. Security badge. And what that does is it basically allows these 2 carbon molecules to get into the citric acid cycle. If you notice, this CoA goes away, so it basically just drops them off. So it acts as like a visitor tag. So, if you went, if your parents come to Hardin Valley for a reason, they check into the office, they'll get a little sticker that they put on their shirt. That's what the CoA does. But the nice thing is once they get through the door, the sticker leaves. Now that doesn't happen with your parents, they have to wear it in the building, but that's kind of the idea. That's what that CoA does for us. As we turn, we get out our 2 CO2s right here. We get out some more buses. We generate a little bit of ATP, and we get out an FADH2. So that's, in a nutshell, what happens there, okay, as we run the citric acid cycle. So pyruvate comes in, we take a carbon off, we get off some electrons. Anytime you lose a carbon, we get electrons with that. CoA attaches, brings it into the citric acid cycle, or also known as the Krebs cycle. Okay. And then it drops those 2 off. We make 2 more CO2s. Every time we get carbon off, we get some electrons off. In this case, we get some energy. Okay, so that's kind of the gist with that. All these slides do is just break it down. I just want, and it's way complicated. I know you look at it and go, oh my gosh, a lot of names, a lot of intermediates. Don't care that you know those. You can go look at this picture and be like, oh, that's crazy. Again, the highlights are in there. Go back to that front page and look. So the last little bit I just want to talk about, and we'll wrap up. In the mitochondria, electrons from the NADH and the FADH2 pass down the electron transport chain. So it does that, and that way we can pump some hydrogen. Okay, so we want to pump that hydrogen into that. So as it does that, that hydrogen creates a concentration gradient that we then can use for ATP synthase. So that's basically what this says, much like we looked through. And so you can take a minute to write this down or read over this. I want to talk about the picture because that's the most important thing. So let's hit this picture right here. So you've seen this picture before as we go through. So let's just kind of address some of the things. So here's our bus. We're going to drop off some electrons. Here's another bus. We're going to drop off some electrons. So here we've got some empty buses. I'm just going to call them EBs. Okay. As the electrons pass, okay, the electrons lose energy. Okay. And it loses its energy because we are active transport for hydrogen ions. So we're moving these hydrogen ions across the membrane. Okay? So we're going to have high hydrogens here. We're going to have low hydrogen here. Okay? Now once that's done, the hydrogens then, through facilitated transport, diffusion, okay, will move down through here. Lost my color. Move down through here. Okay. From natural high to low, causing ATP synthase to turn and generating a whole bunch of ATP. So the more hydrogens we move, the more ATP we make. The more electrons we drop off, the more hydrogens we move. So we constantly have that process. So now you should be asking, what happens to our electrons? Well, they are empty. They don't have any energy. They've dropped down in that ground state. Oxygen comes along and says, hey, I like electrons. Give me some electrons. So it takes them, it finds some hydrogen that's floating over here, and it picks up. And again, that helps it keep a low hydrogen, and we make some water. So that's what we've done there. So we've made our water. We've made our ATP. We can take that ATP anywhere in the cell at this point and do cellular work, is the idea. So if you remember back to the chlorophyll, the chloroplast coloring sheet, the last part of that coloring sheet, which I'm going to give back to you this week because I have it done. I got to get it graded. It's graded. I got to get it in Aspen. Had ATP on there. We talked about the triphosphate versus the diphosphate. Okay, and so that in a nutshell, I know it was a fairly longer video, but that kind of runs through the nitty gritty of cellular respiration, the three stages, the three main stages. Feel free to rewatch. We'll spend class time the next week or so working through this and analyzing this, adding to this process, and I will see you all later.